Alright, after washing your artery 3 times with water for 10 minutes each, we will get rid of all the excess water right here. And then wash it in 70% ethanol. So you want to cover it in ethanol and put it back on the shaker for 30 minutes. After 30 minutes, grab your artery from the shaker and add new ethanol. Just does not matter. Get rid of all this ethanol, add new ethanol, and we're gonna let it sit in that for a week. And in this case, you don't need to come and change it. That is it.
So right now what we do is we want to measure the contents of like the DNA contents in your fresh samples and your cell samples. So we already like analyzed them last night. So basically it's ready to use. So we will start from the third step. .200 microns air buffers to both tubes. And then we'll add 200 micron acenos and mix again. And this is called DNA mini spin columns. So we pipette our mixture from last steps in here. And the white stuff here is actually the membrane. Like we use this to separate the DNA contents from other parts. Label which one is the cell and which one is fresh, since we need to centrifuge them. For each samples, you will have about 700 micron. Right now, we will spin these spin columns for 1 minute at 8,000 RPM. So, after these steps, theoretically, every DNA contents is already left on the wet thing like the filter. In here, in both tubes. We'll spin them for another 1 minute at 8,000. In each tube. Ethanol, so we need to spin for longer time and higher speed to dry everything. You pour everything in the waste collection. We will put them into clean tubes. And label what is what. In this way, since you need to elute the things on membrane, so you want to add the waters around, near the wet filters, but please be careful. Do not poke your wet filters in that way. Repeat the same steps for your decel. After incubation for 1 minute, the elution buffers should already dissolve everything in any DNA contents in the elution buffer. Another minute. So after spin for 1 minute, like you can discard the top parts, which is the filter parts, in waste containers. And what we get in here is actually your DNA contents from your fresh samples and decel samples. And this is all for the part A in your lab 2. So right now what we do is we want to measure the DNA concentrations of your fresh and decellularization samples. So before we actually put anything on the nano drops, the first thing you need to do is you need to use the still waters to wash it for 1 or 2 twice to clean the remaining surface if last users left. You can use 1 or 2 mils to just cover the small black dot, and then do something like that to wash it. And these steps need to be repeated for twice. Use a new pipette tip and add another 1 drops the DI waters for blanking. Okay. And here we will use the nano drops, and we goes into the nucleic acid models. You select it, and the first thing you need to do is you blank it. After blanking, you want to make sure whether or not it's truly be blanked, so you need to measure it. You see the nucleic acid is almost 0, it's really low, which means it should be work. And then right now you can wipe away the blanks and add your own samples. And marry.